right, good morning, everyone. Happy Friday the 13th to you. Hopefully, yeah, I know it's a pretty frightening day, and we're going to talk about some pretty frightening organisms today. At least start chatting about them. Before we get into that, just a couple of things to kind of keep on your radar. Been looking at the syllabus. You know, we have our first exam 2 weeks from now. Where we're going to go from here, remember Monday, Wednesday, I'm going to put videos online for you to watch, have another couple of quizzes for you to follow along with as well. We won't meet again until next Friday. Then the week after that, the week of the exam, what we're going to do is on that Monday, well actually, we're not going to have a lecture. I'm going to bring to class kind of a couple of questions that I think help synthesize everything we've been talking about the last couple of weeks. We're going to break you guys off in groups, have you work on these, and we'll have a little bit of a discussion on what you came up with. The idea being a lot of what we're going to be doing in that class is what's going to be expected of you for the written part of the exam. So we're just really getting you going for that. And then the Wednesday, before the exam, just like every other class before an exam, we're just going to meet anyone who wants to meet. We'll be here, we'll discuss any of the content that you want to, go over anything, questions you would have, so that you're prepped for that. And also what I'll do, probably start about next week, I'll pop up a practice exam, pretty much give you the written exam from last year, so you have an example of what that looks like, as well as a couple example multiple choice questions, so you at least, you come in at least knowing what to expect, in terms of those types of questions. So just giving you a primer of what to expect for the next 2 weeks. But what we're going to do for today and all of next week is talk about spoilage organisms. And starting with the aerobic spoilage organisms that I put in parentheses, because we're going to have a couple of diversions today, in which most of what we'll talk about is the organism Pseudomonas, which is a strict aerobe. It's really the classical example that we use when talking about aerobic spoilage organisms. You'll see why it can cause all sorts of issues with foods. But we're also going to delve into a couple of other categories: the lactic acid bacteria, the thermophilic bacteria, and the yeast and molds. So again, as I mentioned in the first class, put up a big list of all the microorganisms that have been associated with foods in the past, and it's a list of well over 100. And say, clearly we don't delve into each of those. We tend, when talking about spoilage organisms particularly, to look at them as these broad categories. We'll talk about why in just a second. But we will, for each of these, try to use some specific species as kind of an example of what a predominant spoilage organism in each of these categories does to foods. So when we talk about aerobic spoilage, we're simply meaning what are the organisms that you take some food, you blend it up, in some buffer, and you put it down, and you plate it onto this media that's just generically called aerobic plate count agar. If you're taking a food, if you're taking Food Science 409, undoubtedly you have or will use this media. Just a combination of things such as tryptone, which is really just a mix of amino acids and small peptides, yeast extracts, which provide some carbohydrates, some vitamins, dextrose, another word for glucose, some agar to solidify it, water, to dissolve everything. And basically, it's what organisms have the ability to grow on this very basic media, and pretty rich media. It has a lot of carbohydrates, amino acids, other things that bacteria need in order to grow. So we're also supporting the growth of a lot of microorganisms that don't have the ability to synthesize a lot of these components, amino acids and otherwise. This thing right here, if you're doing anything microbiologically related with your careers as you move on, this is something you're going to have as a reference right at your desk or on your computer. The FDA's BAM, or Bacteriological Analytical Manual, you can Google that and pull it up. really big PDF. It's also a document that is updated on almost a month-to-month basis as things change, as new information comes in. But it's everything that you would need to know if you're trying to isolate a specific pathogen, specific group of organisms. What are the recommendations that the FDA has in terms of what media to use, how to prepare, that food? Obviously, if you're trying to isolate a pathogen out of produce versus bread versus, pick whatever food, there are going to be different recommendations of how do you extract those microorganisms. So basically, this is, kind of your go-to manual of what are the protocols that actually have some really good scientific basis for working. But additionally, we talk about these broader categories because of the types of growth that we get when we change the pH of foods or the atmosphere around a food or the temperature. We kind of had a little bit of an intro into this, in which this is an example using produce. Again, just taken from a microbiology textbook, saying that most foods, if you have a pH of greater than 4.5, again, that kind of cutoff for bacteria, you're storing it in the air, refrigeration temperatures. The predominant spoilage organism that you're going to see are pseudomonads, or again, these aerobic, spoilage organisms. Not to say you don't see any of these other ones, but it's just, again, these are good rules of thumb for, taking a look at a product and saying, what do I think the issues might be? We take that same product and we now reduce the pH below 4.5. And in this example, they're saying, we can also throw in a little bit of temperature abuse. Maybe it wasn't always stored at refrigeration temperatures. Those acidic conditions are now going to suppress the pseudomonads, that don't do very well in those conditions. And now we're going to start to see fungi, so yeast and molds, and even some lactic acid bacteria, that again we'll talk about next time, start to propagate. So we can see, this is kind of a general theme that we would see is, reduced pH is going to encourage the growth of these types of organisms. We take a higher pH. We now use modified atmospheric packaging, reduce the oxygen, increase the carbon dioxide. Again, your aerobic organisms, such as your pseudomonads, are now going to be suppressed, and it's going to allow the growth of organisms such as Erwinia that fall into a bigger category of bacteria called the Enterobacteriaceae. Erwinia is an organism that naturally occurs on the surface of plants. So really all you're doing, just like with selective media in the laboratory, where you have hundreds of organisms, you put them down on a plate, but that media has antibiotics, has other things that select for a small group of microorganisms. Pretty much doing the same thing here. By modifying the food, by having a food with a particular type of characteristics, modifying the atmosphere, you're selecting for these types of organisms. Taking the same environment and again reducing the pH, and again we get the fungi. In this case, they're just saying the yeast and lactic acid bacteria that propagate. So really just thinking about, what are the basic conditions that will allow the propagation of the organisms that we're going to talk about today? It's higher pHs and aerobic conditions. So that was on vegetables. Looking at the same thing on meats. Again, we're going to see some common themes here. Again, take meat, poultry. This is fish. Here on the right. Just look at meat and poultry right now, stored under aerobic conditions, and again, it's the pseudomonads that are going to propagate. And I think as you'll kind of see as we talk about the pseudomonads, these are organisms that are just really good at growing on various substrates such as this. But again, modified atmospheric packaging, increased carbon dioxide, even with some oxygen. The carbon dioxide suppresses the pseudomonads, and you get this organism we talked about before, Brocathrix, that tends to propagate. This is an organism that's naturally found on meat surfaces. It's a gram-positive. Actually, it's somewhat related to the foodborne pathogen Listeria, but it's not a human pathogen. Brocatrix is, a facultative anaerobe. It, grows pretty well under these conditions, but if you take the oxygen away, now we're going to get, mostly growth from the Enterobacteriaceae and the lactic acid bacteria. So again, lactic acid bacteria are really going to, do well in the absence of oxygen, and as we showed in the previous slide, particularly when the pH is lower. They do really well when we have 100% carbon dioxide, and under vacuum packaging, it says on meats, we'll tend to see the Pseudomonads and Brocatrix as well. Remember, with vacuum packaging, we're removing a lot of the air. We're not necessarily removing 100% of the oxygen, so Pseudomonas can do fairly well. With fish, we see some different organisms, but similar, themes. We've got the Pseudomonads, lactic acid bacteria here at the bottom. We also see these two other organisms that pop up, Shionella and Photobacterium. Just want to take a little bit of a segue, because I think those are, interesting, specific, examples that are interesting to talk about. But again, in the context of where we're going. So again, slide, a lot of information in here. This is, the details aren't important, but I just want to kind of use this again as a way of introducing these organisms that we're talking about today. So again, a table taken out of a standard food microbiology book, asking the question, we take sterilized beef, so this was done in the laboratory, we inoculate it with some of these various spoilage organisms, we let it incubate at refrigeration temperatures, refrigeration temperatures for 30 days. And this was probably done with a sensory panel, give it to people and say, what do you detect? You can obviously see, Brocatrix, buttery, slate yeasty, things like that. These are obviously things that you're not anticipating, with fresh meats. We talked about Carnobacterium in the previous lecture with modified atmospheric packaging. These are the guys, Lactobacillus all the way down to Leuconostox, that are the lactic acid bacteria that again we'll go into in more detail. And at the bottom, the Serratias, that can also be found on meat products. These are good examples of Enterobacteriaceae as well. So Enterobacteriaceae again is a group of several dozen different species. All I really want you to take away is the big view of this, to note some of the common defects that you see. Buttery, acid, sulfurous, because these are ones that we're going to specifically show you why these defects form. So one of the things I hope you come away with in the next couple of lectures is not only these group of organisms, some of the defects that they cause, but also showing you, there's a physiological reason that the bacteria are doing these things. The bacteria, they're not growing on food because they're looking to spoil it. They're growing on food because it is a wonderful source and they appreciate you providing it to them. But sometimes as some of their metabolic byproducts or end products are some of these compounds that we detect as being objectionable. So Shionella and Photobacterium, the two that were mentioned in the previous slide, these are both gram-negative organisms that are particularly good at producing this compound called trimethylamine. Photobacterium does this much, much better than Shionella. For anyone who has smelled that awful aroma odor of spoiled fish, that is what you are smelling. So these are the predominant organisms that are associated with spoiled fish. Okay, so let's now kind of delve into more specifics on the organisms we're going to talk about today. So hopefully, one thing to point out with these guys here, the Shionella Photobacterium, a lot of spoilage organisms, where do they come from? Well, they were naturally present in the food in the first place. Shionella Photobacterium are marine organisms, that are naturally going to contaminate, fish meat, when it's being processed. It's going to be naturally present on the skin, of the fish and such. Likewise, here's a graph with an example of bacteria that have been isolated from cut pineapples. And again, so this is cut immediately, sampled. We have yeast, we have lactic acid bacteria, other groups that they call mesophilic, bacteria that are naturally present, and again, at fairly high levels, on these foods. So again, what we're really doing is, by the process of how we process a food, how we store it, the long period of time, based on the conditions of that food, we're selecting for various spoilage organisms. Raw milk, if you were to take it, and just plate it out, it's usually about 10 to the 4th to 10 to the 7th APCs, which are aerobic plate counts. So again, the idea being these spoilage organisms are often things that were naturally there that are just able to grow over periods of time. So we're going to use Pseudomonas as an example of an aerobic spoilage organism. Not only because it's got a lot of characteristics that make this, I think that demonstrate what these spoilage organisms can do very well. But also, honestly, as you saw from the meat slides, from the fish slides, from the vegetable slide, it's a fairly common spoilage organism. One of the reasons is it's ubiquitous, so a word meaning it is present in a lot of different environments. As hopefully we'll see in this, Pseudomonas can do a lot of things. It can generate energy from a lot of different compounds. It can gather up compounds from the environment fairly well. Most of what it does is growing by the Embden-Meyerhoff-Parnas pathway, which we've talked about before. That's kind of the glucose down to pyruvate, and then it metabolizes pyruvate through the TCA cycle. It's really good at growing in food products, but as we'll see, primarily what it's doing is producing a lot of different enzymes that allow it to break down food components, making the foods highly perishable. And specifically, we'll show you how this organism is really good at doing three things: proteolysis, so breaking down proteins; lipolysis, breaking down especially triacylglycerides; and amino acid degradation. So this slide, again, the details are not important, but the concept of just showing you that, Pseudomonas, one of the reasons it's so successful, besides the fact that it's ubiquitous, is it has the ability to use a lot of different things as a carbon source. So what this slide is basically they've made some laboratory media that was designed to mimic, a meat surface, providing Pseudomonas, particularly this species, Pseudomonas fragii, with a variety of different carbon sources that are all shown in the different, styles of, triangles, circles, boxes, and otherwise. And then they grew this organism in that media over time, so that's the x-axis, and basically asked, what's the concentration of each of those carbon sources? So these are things such as lactic acid, which is naturally going to be present in meats as the rigor process happens. Some other things such as D-glucose, D-gluconate, glucono, delta-lactone, which is just another carbohydrate that can be found, creatine, creatinine, and aspartate glutamate. So again, specifics aren't important, but these are just various types of carbon sources that are found on meat products that not all microorganisms are able to use for growth. With this example, that curve right there is the growth of Pseudomonas. And I think you can see with all of these carbon sources, eventually they start to drop off, meaning Pseudomonas can basically take a look at this smorgasbord of things and say, I can utilize all of these things for growth. This is just, again, an important trait of this organism that makes it so successful as a spoilage organism. But also, kind of what you see in here, and this is a term you may remember from, hopefully they taught you in Micro 201, dioxic growth. This is that biological phenomena where if you provide a microorganism with a whole host of carbon sources and it's able to use all of them, like this example here, it doesn't necessarily use all of them simultaneously. Often there may be one or two that are preferred that it'll start, using, and once that compound becomes exhausted, then it'll start synthesizing the enzymatic machinery to use some others. And you see that here, in which, predominantly Pseudomonas really likes to use the L-lactic acid, that is in the meat product. And once the concentration of that starts to drop off, then it starts to use, some other compounds here, including the creatine and the creatinine. So we've got one compound here, these other two down here that also seem to be used simultaneously. But again, it's that idea that not all the carbon sources are going to be utilized at the same time. Okay, so what are the things that make Pseudomonas, besides the fact that it can use a variety of carbon sources? And the one thing I didn't touch on, but I think you kind of saw that in some of the earlier slides, is it's also a psychotrophic organism. So it grows pretty nicely, at temperatures that we tend to store foods at. I'm thinking like room temperature, or slightly elevated. But also when we put these foods in the refrigerator, it's certainly going to slow its growth, but it's not going to stop it. So one of the characteristics of Pseudomonads is they produce a large number, two things. So first of all, they produce a large number of proteases. So these are going to be enzymes that are synthesized in the cytoplasm and are secreted into the exterior environment, and basically will go around and chew up any proteins that are in the environment down to short peptides or free amino acids. We mentioned before when talking about milk and nutrient availability, said that most of the amino acids that are present in milk are tied up in these large proteins called caseins. Bacteria can't use large proteins immediately as an energy source or a carbon source, so they need to find ways to break proteins down into free amino acids or short peptides in order to be able to transport them across their membrane and to be able to directly incorporate them into new proteins or break them down for energy. So Pseudomonas can produce these proteases. To say that they're optimally active at temperatures you would expect for most bacterial enzymes that we talk about, around 30, 45 degrees, 37 being the optimal growth temperature of many of these organisms. They're what we call metalloproteases, meaning they require usually a divalent cation in order to function. So the issue is not just the fact that they produce these, but that these are extremely heat-stable enzymes. The table that's shown in the bottom right is basically demonstrating this, in which these researchers isolated some of these proteases from a Pseudomonas, the Pseudomonas fluorescens that we've referred to before. They've subjected them these proteases to a number of heat treatments, one being the control, so again, no heat treatment. By definition, it's going to be 100% relative activity. That's the activity that we're comparing everything to. If we heat those proteases 65 degrees for 30 minutes, again, coming back to vat pasteurization, you only lose you still retain about 14% of the total activity of those enzymes. So this is a time and temperature that enzymes from E. coli or some of our other gram-negative organisms are going to almost fully denature at that time and temperature. So this is fairly remarkable that these enzymes persist after this treatment. 72 degrees for 0.25 minutes. Again, we'll come back to that's high temperature, short time pasteurization. All the milk and most of the milk in the grocery stores and the creamery, that's how they've been treated. After that, we still have 30% activity that remains. And you can go all the way down and even see at boiling, 1 minute, 3 minutes, 5 minutes. We still have a pretty good clip of enzymatic activity that survives. So what does this mean to you if you're, I guess you could say, a dairy, a producer of dairy products, is, well, you're harvesting, milk from animals. We know that one of the first things you want to do as quickly as possible is to refrigerate that milk, because you're trying to prevent the growth of any microorganisms that may have contaminated the milk from the animal, from the environment. One of the things that you're really trying to do also is prevent the growth of any pseudomonads that are in that product. Because what this means is if you allow any growth, it doesn't matter what you do to that milk afterwards, vat pasteurization, high temperature, short time pasteurization, you're going to kill the microorganisms, certainly. Pseudomonas is not generally it's not a heat stable organism, but its proteases are going to persist. And these proteases, we say, are still active at refrigeration temperatures. So as you store the milk, these enzymes are going to start, continue to chew up the caseins that are present. We say these have a broad substrate specificity. Just again, another biochemical way of saying they don't. They tend to target a lot of different proteins. They're not specific to any particular ones that occur in foods. So what are the ramifications of having proteases that are chewing up the caseins in milk? So we mentioned before, caseins are present in the casein micelle in milk. That basically it's surrounded by a membrane. Is where these various proteins called alpha caseins, beta caseins, and a little bit kappa casein as well are contained. And if we look at the structure of just one of these, so this is beta casein, in which is 210, 211 amino acids in length. So each one of these colored circles is representing a single amino acid in this protein. But they've been colored to indicate what category of amino acids that each one of them is. So when you're taking biochemistry and you're learning about the amino acids, you've learned we can group them in various ways. Branch chain amino acids and sulfur-containing amino acids and such. This has been categorized into the amino acids that are hydrophobic and those that are hydrophilic, and also ones that are positively and negatively charged. Things like glutamic acid, glutamine, aspartic acid, asparagine are going to be examples of charged amino acids. Hydrophobic amino acids are going to be things like prolines and leucines and things such as that. So the exact amino acid composition isn't important here, but I think what you can see, taking big picture look at this, is a fairly good clip of amino acids that are present in beta casein and other caseins are yellow or hydrophobic. Okay, so interesting little trait, but what does that exactly mean when it comes to proteases that are chewing up, caseins? And it has to do with the fact of how our bodies perceive strongly hydrophobic short peptides. We tend to perceive these as being bitter compounds. So as caseins get chewed up from these larger proteins into small peptides, 2, 3, 4, 5 amino acids in length, and those peptides are highly hydrophobic, this is one of the things that will make spoiled milk start to taste a little bit bitter. So again, you want to keep pseudomonads out of your product for these various reasons. But it's not simply stopped there with bitter peptides being generated. As I said, these are pretty powerful proteases. They have a broad substrate specificity. They can continue chewing these caseins down to their individual amino acids and release these compounds. We'll see, particularly when we talk about the lactic acid bacteria, I think in maybe the fermentation lecture, certain amino acids actually have some defining flavor characteristics. The ones we're specifically talk about today are the sulfur-containing ones, such as methionine. So again, details of this slide are not important, but the bigger concept of what's going on is. So there's also, bacteria that can be present in food products that have the ability to break down amino acids. Again, they're not doing it to make the food taste bad. They're doing it because it helps them, utilize these amino acids for energy or for carbon, other things that help them grow and divide. There's two different ways that bacteria can generally, break down methionine. A couple examples, of organisms that do it fairly well are shown here. One of them is a two-step process using an enzyme that's called an aminotransferase. In this case, it's methionine aminotransferase. That will basically deaminate the amino acid, followed by a second reaction in which we decarboxylate it. And you form this end product called methane thiol. So methane thiol, is basically a single carbon linked to a sulfur group. That, as we see, in and of itself, this compound has a very obnoxious, odor to it, but also chemically, it can convert to two different compounds, dimethyl disulfide, dimethyl trisulfide. So really just, the steps aren't important, but these end products, I think are what are important to key in on right now. On the left-hand side of this slide is simply showing there's a second mechanism by which, a certain bacterial species can generate methane thiol. So what are the implications of this? Many of these sulfur-containing, small molecules that are generated from sulfur-containing amino acids, methionine, cysteine being, the two, have, flavor aromas that we perceive, as being off flavors of, off aromas. Starting with the top, hydrogen sulfide, which comes from cysteine, is kind of that defining sulfur characteristic of rotten eggs. Here was methane thiol that we just talked about, which is perceived as kind of cooked cabbage, fermented cabbage. So think of cabbage that you boiled, that type of aroma. Noting the odor threshold of these. So these are in parts per billion, an extremely small amount. We have the ability to perceive these at extremely low levels. So it doesn't take an awful lot, to generate these compounds to get to a point in which we can perceive them. We mentioned, some of the sulfides in that previous, slide. Dimethyl, dimethyl trisulfide and the such. Again, being, cabbage, sulfur, garlic, types of, aromas. Again, not going to ask you to match compound with aroma, but recognizing, how and why, these are forming. So I kept kind of the right-hand side of this slide, again, just to make a point that we've made once, again, we'll make it, a couple times this semester, is these are not only spoilage, or compounds that are associated with, spoilage, they're also associated with some cheeses, and these are, found in Limburger. Yeah, Limburger. If anyone has had that, you know what that is. That is a really. Some people think it's wonderful. Some people think. I think the majority of people think it is a really awful smelling cheese. But everything, things that are much more recognizable, cheddar cheeses, parmesans, and the such. So keeping in mind, particularly when we start talking about fermentations, we say, what is a fermentation? But it's really controlled spoilage of products. So instead of allowing milk to get to a point in which we produce, so many of these compounds that we can't consume it anymore, we're controlling the formation of these compounds because they're actually at a lower level are considered to be beneficial flavor, or contribute to the beneficial flavor profile of many fermented products, again, specifically in cheese. So I always call it fermentation, controlled spoilage. There are other people in the department who choose to use the term controlled rotting of foods. Just for completeness, we've given you one example of how amino acids can be broken down to form flavor compounds. There's also a host of enzymes that bacteria can produce that are called amino acid deaminases. That all they do is take an amino acid and they remove the ammonia group from it. So from a flavor profile standpoint, again, ammonia is not something that we generally would perceive as being positive, again, except in some fermented products. From a standpoint of why do these enzymes exist in the first place, there's really two reasons you can think of. One is, in this particular example, where we deaminate serine, we produce the compound pyruvate, which is one of the compounds that we can use to feed right into the tricarboxylic acid cycle to generate energy. Additionally, you generate ammonia. I think ammonia is a compound with a high pH. So if you're a bacteria that's in a high acidic environment and you want to try to figure out ways to bring the pH back closer to neutrality, you can basically start deaminating your amino acids to generate ammonia for this purpose. Okay, but I also wanted to take that as just a segue away from the Pseudomonads just for a second. To also discuss where else we see some of breakdown of amino acids and where it impacts not so much the flavor profile of foods, but how, if you will, how we react to them. So this is a amine formation that is particularly associated with fish. And this is a process where the bacteria that are present and growing on the fish are taking amino acids and they're removing the carboxyl group. So they're basically decarboxylating them. A couple of examples up here, histamine is this compound here, and I think if you compare it to the structure of histidine, you'll see that there's a carboxylic acid group that's missing right there. Putrescine, cadaverine. Yes, I was talking that these are more from a health impact, but I think you can tell by the names. These are also nasty smelling compounds as well. These are decarboxylated ornithine and lysine. So these are the three compounds that we most often associate with this particular phenomenon that we're talking here. And that phenomenon is called scombroid poisoning, which is often will occur in certain individuals who are sensitive when they've eaten spoiled fish that has begun to spoil, and certain bacteria have begun to form these three different compounds from their associated amino acids. We perceive it, get dizziness, you get itching, you get a burning, sensation. Basically, it's kind of like an allergic, types of reaction. It's not across all fish, but particularly fish that are of the Scombroideae, family, mackerel, tuna, and the such. And that's the whole point of this table here at the bottom right, is showing you, some of these fish species that naturally have a high concentration of these particular amino acids. So the substrates are there, the bacteria are there, the situation is ripe for producing these amine compounds. Histamine is usually the one that gets most associated with scombroid poisoning, but we recognize that it's not it's necessary for these symptoms, but it's not sufficient. You do need some of these other amines as well. Pointing out cadaverine, as we mentioned, this is a a compound that doesn't smell very well either, and often is used as an indicator of spoiled fish. So some folks will actually do a chemical analysis of fish, and if they can detect this compound, it's considered to be spoiled. Couple of bacteria that are known to do this, but we'll revisit this also when we talk about the lactic acid bacteria, because there are lactic acid bacteria that can do this process as well of decarboxylating amino acids. So again, just kind of using this as a way of demonstrating, besides spoilage, there are other reasons that we're concerned about bacteria that can degrade amino acids that are found on our foods. Yeah.
So you don't need all three to get scomboid, or do you?
You don't. Yeah, they all contribute to it, but you don't need all three. Yeah. Okay, so keeping with the theme again of this is what happens, this is how it impacts us, but again, why would the bacteria do this in the first place? Because again, they're not doing this because they want to make us sick. They're doing it because they want to grow, and we're just collateral damage in that process. So you decarboxylate amino acids. Again, it could be to help break down amino acids so that they can be fed into other pathways for energy. But another really interesting reason why bacteria do this is, again, we talked about you can deaminate amino acids to help raise the pH of the media. You can decarboxylate amino acids to help raise the pH of your cytoplasm. So there's a lot of details on this slide, but we'll kind of go through the important points of it. If we take the amino acid glutamate, so this is the upper right part of the slide, and what we're basically showing is a decarboxylation reaction. So you can see in the reaction, you get rid of a carbon dioxide. You're removing that group right there. You produce a compound called gamma-aminobutyric acid. Looks something like this. Structure isn't terribly important, but what's critical to look at with that reaction is the top of the arrow. You see that in order to complete this decarboxylation reaction, you actually consume a free proton. As you know by the definition of pH, pH is basically the concentration of free protons. High concentration is going to be a low pH. So keep that in mind. A decarboxylation reaction consumes a free proton. So what bacteria do, and this is kind of that diagram that's shown on the left, they say, well, during and the bacteria find themselves in an acidic environment, you can get, where free protons, just by concentration, might be able to cross the membrane. The bacteria, are basically fighting a very large concentration gradient between the inside and the outside. But the bacteria, by importing, amino acid, in this case, it's by an internal membrane protein that's called ADIC. They bring it into their cytoplasm. They now decarboxylate it. So in this case, arginine, diagmatine, which is shown, it's the other reaction shown at the bottom here. And in that process, they basically stick a free proton onto that molecule. And now they can secrete the end product. So you can think this is kind of an indirect way of getting rid of free protons out of your cytoplasm. And interestingly, the way the transport process works is, the energy that is used for transport is basically done by coupling the import of the amino acid with the export of the product. So this is a really efficient way, decarboxylation would be a really efficient way for bacteria to increase the pH of their cytoplasm to help them survive or persist in highly acidic environments. Okay, so we've seen that Pseudomonas can use heat-stable, fairly persistent proteases as a way of generating spoilage compounds, be it bitter compounds, sulfur compounds, others in, and again, pretty much the example we used here most of the time was milk. Lipases, pretty much the same story. So this particular table, these investigators purified some lipases that again are produced by Pseudomonas and are secreted out into the media. Subjected them to a variety of heat treatments, and here they're actually calculating a d-value. So I think pointing out that d-values can not only be calculated for destruction of microorganisms, can be for destruction of enzymes if you so well choose. In which they were spiking the purified lipases from Pseudomonas either into SMS, which is synthetic milk serum, so basically making milk in the laboratory, or just plain phosphate buffer, so kind of a buffer that you would make in a laboratory experiment. Showing that again, if we were to go to maybe 70 degrees, or so, temperatures that are a little bit more are standard for pasteurization, that in this synthetic milk media, you're losing 90% of the activity of those enzymes about every 7.5 minutes. So again, putting that into perspective, at that temperature, most enzymes for E. coli would be fully inactivated within 15 seconds or so. So these are enzymes that, again, if you allow Pseudomonas to grow in raw milk, produce enough of these, you're not going to get rid of them by pasteurization, be it high temperature, short time, that pasteurization, or the such. And again, I think you can really see this when you look at 100 degrees Celsius, and you see again, these enzymes have a fairly persistent, persist fairly well. The other point of this table is also showing, we talked about how d-values for inactivating bacteria. You can take the same bacteria, harvest it from lag phase, from stationary phase, from 30-day, that we referred to as long-term, survival, stage, and they're going to have different d-values. Enzymes are often, their kinetics of inactivation are often going to differ based upon the media that they're in. So basically, simple phosphate buffer, the d-value is going to be dramatically different from what we see if we put these enzymes in something that's a little bit more reminiscent of milk. Okay, so just one last point, before we, because we'll use this as a segue to where we'll begin on Monday, is so what's the big deal with producing, fatty acids? So we'll see that sometimes fatty acids can have, flavor profiles by themselves, but also they can react with, various, alcohols that are naturally present in foods to form, off flavors and off aromas. So this is a chart that has, everywhere from 1 carbon to 10 carbons, representing the fatty acids, all the way from 1 carbon to 9 carbon alcohols, and basically giving you a picture of what flavors form when these, react in chemical compounds, that are referred to as esters. In particular in milk, the two that occur most commonly, because the fatty acids that are most associated with milk fat are butyric acid are basically 4 carbons and 6 carbons, and they react with ethanol that are naturally formed by the bacteria. And the compounds that result are ones that we perceive as having a pineapple aroma to them. So let's actually leave it there for now, and we'll wrap up with the next lecture showing you basically where the ethanol comes that allows these compounds to form. So again, have a good weekend, and we will see you in person next Friday.